I'd like to start by thanking Max and the organizers for inviting me here to share with you some perspectives on topical prophylaxis. And I think particularly it's pertinent because when I think of Botswana, I think about the amazing contributions that have been made in terms of research, whether it's in mother-to-child transmission or even in the PrEP field, where the CDC study was one of the really important studies to show that PrEP works in heterosexual populations. So it's a great pleasure to be with you here today. What I want to do is just share with you some perspectives on microbicides and their potential in terms of an overall plan for combination prevention. So let me start with some epidemiology and share with you the epidemic that we face on a daily basis. So these are data that come from 22 schools that Kuresha is doing a trial in. And in these schools, she measures on a regular basis, once a year, the HIV prevalence in all school kids in the grades 9 and 10. And we're measuring incidence rates in these school boys and girls. So if we look at the HIV prevalence rates in the boys in these schools, and these are fairly large numbers, I didn't put the ends in, but in the boys below 14, the prevalence is 1%. And as we go up even to the oldest boys, who are 19 and 20 years old, we have prevalence rates of around 1 to 1.5%. So this is the sort of nature of the HIV epidemic that we see in young boys. Now, if we look at the situation in girls, what we see is a dramatically different picture. So when we look at girls who are 14 and below, they already have a prevalence rate of 2.2%. When they get to 15 and 16, it's 3.6%, 17 and 18, 7.9%. By the time they reach 20, it's 16%. So that's what we face as the epidemic unfolds in our setting. So if we want to talk about stopping the epidemic or slowing the epidemic, we've got to deal with this problem of these incredibly high HIV incidence rates in young girls. And it translates into a whole range of different features of the HIV epidemic. But in the same community, we have 7 primary healthcare clinics. This is the prevalence of HIV in antenatal clinic attenders, women attending for their first visit for prenatal care. So if you look at in these 7 clinics, over this period, we monitor this on an ongoing basis from 2009 to 2012. In the 16-year-olds and below, we're talking about 8.4% prevalence already. By the time you get to 20-year-olds, 1 out of 4 walking into the clinic for prenatal care has HIV infection. By the time you're 22, it's 1 out of 3. By the time you're talking about 24-year-olds, it's close to 1 out of 2 women walking in for prenatal care already have HIV infection. So we've got to do something about this, and we have been trying in all different kinds of ways to do it. But is this unique to KwaZulu-Natal? Is this unique to Vulindlela? Well, if we look at the UNAIDS data and look at the prevalence of HIV infection in young men and women between the ages of 15 and 24. In the red bars, I show you the prevalence of HIV in young women. In the blue bars, the prevalence of HIV in young men. So if you look at South Africa, Lesotho, Swaziland, Zimbabwe, Namibia, Zambia, all the way to the Central African Republic, Kenya, across the board, we see the same picture of much higher HIV prevalence rates in young girls than in young boys. And this is partly a reflection of the age partnering pattern. And we know certainly in our community that on average in these young girls in their teen years, their partners are about 5 to 7 years older. So what do we understand about this? Well, I have to tell you, actually not much. And from the behavioral studies that we've been trying to grapple with in these schools, what we've found is that there's a lot of pressure to have sex at a young age. In fact, if you come to the 7 clinics in Vulindlela, 1/3 of all prenatal clinic attenders are teenagers, giving you some idea of the very high rates at which sexual debut occurs and in which risky sex is taking place in teenage girls. Some of this sex is for money or for favors, peer pressure. There's a lot about having an older man looking after you. There's a certain amount of status that goes with this. There's a whole range of different features about what drives this kind of behavior. And there's very poor internalization of risk. Knowledge of HIV status is low in these young kids. And there is a sort of view that AIDS affects other people. They don't see it as affecting them. And many of them have really no sense of future as well. They don't think about, what are my opportunities going to be? I'm going to get a job. I'm going to go to university. They don't talk in that way. They don't live in that kind of way. And many of them change partners on a regular basis. They move from one partner to another. Not many of them have multiple partners at the same time, but there are some that do. So when we look at this kind of population of young women, and we think about the prevention tools that we have, and when I'm sitting in the clinic and seeing these young girls coming in, many of them with babies, with babies who are ill, trying to give them some advice around how to protect themselves. And we think about, as we go through abstinence, be faithful, condoms, circumcision, TASP, ARV prophylaxis, which of these can we really implement that we hope would be protective in this kind of population? From my experience, certainly the ABCCs are of very limited value. Most of these girls are already sexually active. Not much point in telling them about being faithful, because even if they are faithful to the man who's actually going to give them HIV. Condoms, for a young girl to be talking to much older men about condoms, and many of them are trying to fall pregnant as well, is not really a practical option. And circumcision, as you know, protects men directly and women only indirectly in the long term. So that leaves us pretty much with TASP and with ARV prophylaxis as the options that we need to explore. So what have we been doing in terms of ARV prophylaxis and topical prophylaxis of other agents? Well, the microbicide field has been one that has been characterized with people with incredible perseverance. To have survived so many trials that have not shown any success means that you're either crazy or you have some really misguided belief system. Well, I have to tell you, having dealt with these people over the last 2 decades, most of them are just plain crazy. But as we think about the field, and there's much debate when it started, but I sort of view Zena Stein's paper as really the starting point of the debate around methods for women. And many of the original studies that were undertaken using naloxone 9, which was already widely available. And moving on to the second class of candidate agents as polyanions, and a whole range of these studies, of which none showed any protection. And that took us then to the most recent class of candidates, which is the antiretrovirals. Most of them are tenofovir based. So that's the sort of history in sort of 30 seconds of the microbicide field. It's characterized by negative results, flat results, and mostly futility studies, and only one study showing efficacy at this point. So, what have we learned from all of these studies? Perhaps the single most important thing that has become very clear to us is the importance of adherence. And we saw that most clearly in the Caprisa 04 trial, where those who have high adherence, when we look at the use of gel in more than 80% of their sex acts, and this is just based on reported data, there's no objective measures here, We show 54% protection, but in those who have much lower levels of adherence, we show lower levels of protection. And part of this has been one of the challenges about exactly how do you get people to take these products. If you simply make it available, will they come? Well, they don't really do that. You've got to try and make this product fit into their lifestyle and their needs. And so one of the reasons for the Capriso-4 study being done in a quietly dependent dosing strategy was that we really had to find out how these women are willing to use this product, especially in this rural community where we did the study, where sex is actually not that frequent because many of the partners are the migrant men. Many of them are taxi drivers, they're workers in the city, and when they come back in the weekends once or twice a month, that's when the sex takes place. So it was critically important that we listen to the women and that we design the strategy to revolve around trying to fit in, try to marry the science and the community and where these two can come together. And the consultation that involves has been really critical. So what have we seen from all of these studies? If you plot the adherence by the objective measure of drug levels, and you compare that to the effectiveness that was observed in these studies, and I've taken all the PrEP studies and included the Capriso-4 studies, what you see is actually a very strong correlation coefficient of 0.86. And you can see here how nicely they track, again highlighting the centrality of adherence within a trial. Now, I mean, that's not rocket science to tell you if you don't take the drug, it's not going to work. But it might seem like something that is axiomatic, but the reality, it has been a real challenge to get women to use these products because we've been trying to impose on them what we think they should be doing. If you look at tenofovir gel and the data we have within the VOICE trial, it was the only arm that showed some level of effectiveness, although this was not statistically significant, because you got pretty low levels of detectable drug. So if only 23% of the women had detectable drug, we saw 14.5% protection. Well, it's not, I guess it's protection, but it's not statistically significant. In the 004 trial, overall, we saw 39%. And this is just looking at recall. And if you look at the use of the applicators to measure adherence, then we get to 54% protection in those with highest adherence. And if we use the drug concentrations and the proposed correlative protection that we identified within the trial of 1,000 nanograms per ml, then we show 74% protection. And if you do a case control analysis, as was done in IPREX and all these studies, where you don't actually use the control arm, you only use the active ingredient arm because they're the only ones you can measure the drug in, and you compare the drug levels in the seroconverters and the non-seroconverters, they all show pretty much similar levels of protection in the 90% range. So where are we with tenofovir gel? Well, it's a long road. We've completed the 004 study, the clinical study report, and the data has already been submitted to the FDA. We had hoped that the VOICE trial would be confirmatory. Unfortunately, it wasn't. And so we are waiting now for the FACS-001 study, which is a confirmatory study that's currently underway using the same coital dosing strategy involving several sites in South Africa. That study is now complete in terms of its follow-up. They're busy exiting participants. We would expect to see the results of this trial probably in December this year or January next year. And thereafter, if that study shows protection, and it's anybody's guess as to whether it will, given the challenges with adherence, then we have the basis to proceed with licensure. In the meantime, WHO has already started developing guidelines for the use of tenofovir gel, and there's also a global strategy that has been developed. So the key is going to be regulatory approval and thereafter launch, and in the meantime, there's plans for manufacture. The South African government has a royalty-free license for tenofovir gel and has partnered with Cipla Metro and created a company called ProPrevin, which has been given the responsibility for developing tenofovir and to be able to manufacture it, distribute it to the whole of Africa. So those plans are underway. They're busy designing factories and so on to make this. All of this will only go forward, of course, if the FACTS trial shows protection. So there's a lot banking on that particular study. So just in terms of pipeline, to end off my presentation, the ARVs are being put into all kinds of formulations, and there are a whole range of ARVs now moving beyond the tenofovir-based products, using maraviroc, using zinc, using MIV-150, using NNRTIs like dapivirine. So there's a whole range of products that are moving forward in different kinds of formulations. And one of the more popular formulations that's now being developed in all different kinds of ways, with different kinds of antiretrovirals, are the ring formulations. And there's continuing work on foams, and an interesting set of products that are now available, the Silks diaphragm that releases tenofovir. There's also a big push for multi-purpose technologies, and part of this is that women are going to need technologies to protect them from pregnancy, as well as HIV, as well as other STDs. So can't we make something that does all of them? So women may be motivated to a greater extent to take something for contraception, but the same product will also then protect them from HIV and STDs. And so there's this whole big push to develop these multi-purpose technologies. And most of these right now are rings and gels. And the rings have been focusing on combinations that prevent pregnancy, using levonorgestrel, preventing HIV, using tenofovir or other antiretrovirals, and preventing HSV2, again, using tenofovir. So this is the big push that's coming through in the field of topical agents. I guess no talk on microbicides and topical formulations would be complete without acknowledging that Africa also has a very substantial epidemic of HIV in men who have sex with men. We've already heard about the importance of oral prophylaxis in this population. And within trying to understand the HIV epidemic in MSM in Africa, what we see is incredibly high rates from the studies that have been undertaken. These are high rates of HIV prevalence across the board, from Cape Town all the way through to Cairo. So what we're seeing in this kind of epidemic is the need for us to formulate and have new strategies as well to protect MSM from HIV. And currently there's a study underway in the MTN to look at a rectal formulation of tenofovir gel. It's actually the same as the vaginal gel, it's just has a lower concentration of glycerin to reduce the osmolality of the product. So this is currently underway, and this, we hope, will lead to studies that will move the next step into looking at efficacy. Incidentally, we also have pretty good understanding from monkeys that if you insert the vaginal gel, you get pretty high levels of the product in the rectum. And we also know that if it's inserted in the rectum, you get pretty good levels of tenofovir in the vagina. But we don't know whether that applies to humans. And so there's an MTN study underway to answer that question, whether we need to apply a gel to both compartments. And in terms of the gel, it's a product that probably has a very niche area, and it's probably a product that's going to be rapidly overtaken within the next few years, either by rings or by injectables or some other product. But right now it's the product we know that works, and so we are moving forward with it as one of the first products to enter the field and to start paving the way for its successors to follow. So just to conclude, I think we now know that we have proof of concept that tenofovir in a gel formulation at least protects against HIV. We need confirmatory data. We need data from multiple sites, and that's coming from the FACTS-01 study, and we're waiting for those results, and those results are going to be key for licensure. We know that one of the key issues with the topical antiretrovirals is that they also address one of the important gender dynamics, like the pills do, is that they put the ability to control the risk of HIV in the hands of women as a women-initiated technology. And we look forward to new formulations and multi-purpose technologies. And I, for one, am very excited about the prospect of products like GSK 744 long-acting, as I see them as really the next generation of products moving us forward, looking at more practical ways of implementing PrEP formulations. And that we need to find ways to improve adherence. And adherence in trials is very different from adherence in the real world. And I can tell you this because we now have an implementation study where we're making post-trial access available to tenofovir gel. The dynamic and what's going on, and women now being told that this works, as opposed to telling them every month that we don't know that it works, has a very different dynamic to adherence. So one should not extrapolate from clinical trials as to what real adherence will be in the real world. And there are several new products in the pipeline, MPTs, multi-purpose technologies, and all of these, need to be moving forward while we try to progress with tenofovir gel. And let me end off with just leaving this up. I'm just going to read very quickly so that you know that there are many falsehoods and arguments that I've had to have with many people. And I want to put them up here because you might want to discuss some of them. But one of the arguments I've heard, even when I gave a talk at the NIH recently, is, can we really justify using these antiretrovirals, particularly the oral ones, for prophylaxis when we have so many patients still needing treatment? So shouldn't we first wait to put everybody who needs treatment onto treatment, and then if we have spare drugs, we can think about making it available for prophylaxis? Is it really safe to give healthy people these drugs? And are these really effective? We have all these results that show, inconsistency, particularly in women. And healthy people just won't take these drugs. They won't insert these gels. And, in these ARVs, would we be just encouraging resistance and putting future treatment programs at risk, particularly ones that are tenofovir based? And are we simply leading to a situation where we will be, women or men who are using these products will increase their risk by stopping the use of condoms and so on? So I've written pieces that argue against each of these, but I thought I would just put them up so that we get some idea of what are some of the big questions and challenges that we often face as we think about the use of topical and oral prophylaxis. Thank you.